In this tutorial, we will discuss the candidate regulatory element details view. To produce a details view for a candidate regulatory element that I am interested in, I simply click its row in the search results table. The first page of the details view is the z-score page. This page displays the z-scores for all 4 epigenomic marks for the selected candidate regulatory element across all cell types. For the histone marks in cccF, 2 values are displayed in the table. One is the z-score for the mark alone, and the other is the average between the mark z-score and the DNA's z-score. All of the tables are sortable by clicking the arrows next to the column headers, and all of the tables are searchable by typing either cell type names or z-scores into the search boxes. The tabs at the top of the page provide access to other pages with different information about the selected candidate regulatory element. The second page is the Nearby Genomic Features page. The top row of 3 tables presents nearby genes, nearby candidate regulatory elements, and nearby SNPs by simple linear distance. The second row of 2 tables presents genes and candidate regulatory elements within the topologically associating domain containing the selected candidate regulatory element. The tables on this page are also sortable and searchable. Gene links lead to gene cards, and SNP links lead to ensemble. The third page provides a list of transcription factor and histone ChIP-seq experiments in ENCODE with peaks that overlap the selected candidate regulatory element. The center column shows the number of experiments with overlapping peaks, and the rightmost column shows the total number of experiments for that particular factor or histone mark available at ENCODE. Clicking a row in the table brings up a detailed list, which displays the cell types and ENCODE experiment accessions that had peaks intersecting the selected candidate regulatory element. The fourth tab displays RNA-seq derived expression levels for the nearest gene to the selected candidate regulatory element. This tab is only displayed if the candidate regulatory element is within 2 kilobases of a gene. The fifth tab displays rampage signal for the transcription start sites associated with the nearest gene to the selected candidate regulatory element. This view and the gene expression view will be discussed in more detail in a future tutorial. The sixth tab displays candidate regulatory element orthologs in mouse identified by liftover. When searching screen MM10 for mouse candidate regulatory elements, this tab displays human orthologs. Clicking a candidate regulatory element link from the list performs a search on screen MM10 for that candidate regulatory element. Finally, the Signal Profile tab displays a snapshot of the signal for all the epigenomic marks across the candidate regulatory element for all available cell types. The numbers next to the peaks give the maximum signal value at the summit of the peak. The table columns are sortable by clicking the arrows at the top of the column headers. The black ENCODE boxes provide a link to the ENCODE portal for the associated experiments.